Thank you, Scott. So, hi guys, I'm Momo, and this is Karthik. And we have been working with Alpa on techniques of fluorination on 6-thiopurine analogs. So, before I say anything else, I just want to say that our research is really exciting because it's actually been going towards making a real difference in the lives of those with cancer. And just knowing that is really exciting. So, over the last 6 weeks, Karthik and I have been working with purine analogs of an anti-cancer agent called 6-thiopurine, which is especially effective against acute lymphoblastic leukemia. So let me explain to you how the drug works in the body. The inactive form of 6-thiopurine is called azathioprine, as you can see here. The azathioprine, when it goes into the body, it goes through a series of metabolic reactions and is converted into 6-thiopurine. 6-thiopurine is then converted into 6-thiouric acid and into a compound called, okay, ready for this? Deoxy-6-thioguanosine 5'-triphosphate. Yeah. It's also known as DTGTP. So when this DTGTP is present inside the cell, it is incorporated into the DNA, which causes cell apoptosis, and that thus stopping the uncontrollable cell growth that defines cancer. However, 6-thiopurine has quite a lot of side effects, such as pancreatitis, immune system inhibition, and hepatotoxicity. The main condition that we focused on is a condition called jaundice. And as you can see here, some of the symptoms of jaundice include a yellowing of the eyes and skin, which is due to the buildup of a yellow pigment in the body called bilirubin. If it'll click, there we go. All right, so bilirubin is the waste product of the breakdown of old red blood cells, a process that occurs in the liver. So there are two forms of bilirubin. The first type is unconjugated bilirubin, which is nonpolar and cannot be excreted from the body in the urine. It needs an enzyme called UDP glucose dehydrogenase, or UDP-GDH, in order to convert it to conjugated bilirubin, which is polar and therefore can be excreted from the body. So 6-thiopurine and 6-thiouric acid both inactivate the action of this enzyme, which causes unconjugated bilirubin to build up inside the body, and which ultimately results in the condition called jaundice. So the main purpose of our research was to find a way to synthesize a slightly altered version of 6-thiopurine that would have anti-cancer properties but would not have those dangerous side effects. So in order to do this, we looked into methods of fluorination, in which the hydrogen at position 2 or position 8 of the thiopurine is substituted with a fluorine atom. This could be beneficial for a multitude of reasons, as you can see, and this substitution could potentially keep 6-thiopurine from being converted into 6-thiouric acid, which could potentially keep the enzyme UDP-GDH from being inactivated, which would not allow jaundice to occur in patients treated with the drug.
So for methods of fluorination, we had two. The first is the diazotization reaction, which is converting 2-amino-6-chloropurine into 2-fluoro-6-chloropurine. And our second is the Traub synthesis, which converts 4,5-diamino-6-hydroxypyrimidine plus the addition of guanidine hydrochloride to turn it into 8-amino-hypoxanthine. So for the diazotization reaction, we first put in our 2-amino-6-chloropurine that we got in factory settings, and we got 40 milligrams of that, and then we added 40% tetrafluoroboric acid and 1 milliliter of that, and put it in a flask and mixed it for 30 minutes. Next, the mixture was cooled at -2 degrees centigrade in an ice bath. And then we added sodium nitrite and DI water. Then it was cooled to -8 degrees Celsius for 2 hours. and we added 7 milligrams of urea, and the mixture was stirred and heated up at 50 degrees Celsius for 1 hour. So to purify our solution, we had to put it through a silica column, and to do that, we added 50% sodium hydroxide to regulate the pH of our material to about 3 or 4. And then we filtered it through the silica column, like I said. And to create this, we just had a glass column. We put a wool at the bottom to plug it up, and then we added silica gel, and then we added our liquids, which was dichloromethane and methanol in a 9-to-1 ratio. Our next process was to characterize this material, and we put that through a UV spectrophotometer, and this just checks the absorbance of the material by shining light through it, and it reflects it back. And we checked this between the absorbance between the wavelengths of 200 to 400 nanometers. And each material has a different absorbance, so we were able to characterize the material. Next, we used our rotary evaporator to filter out the diluent in our solution. And then for our final method of characterization, we used the proton NMR, nuclear magnetic resonance, and that determines the placement of protons in our molecule to finally characterize it. And each compound, like glass, has different spectra. For Trop synthesis, we converted 4,5-diamino 6-hydroxypyrimidine plus guanidine hydrochloride again to make 8-amino hypoxanthine. So our first step was to combine guanidine hydrochloride and 4,5-diamino 6-hydroxypyrimidine in a 1 to 2.5 ratio, and again, we acquired these materials through factory settings. Next, we added them to a pestle mortar and ground them into a sand-like mixture, and then that was transferred into a crucible and heated at 200 degrees Celsius until it was melted. Our next step was to filter this material, so we added our solution through the top of the flask and added water, and we got our final solution, and the vacuum filtered out all the water. And then we added methanol and ethanol in a 5 to 5.5 ratio, and then we also added diol ether to help evaporate everything. For characterization, we used thin layer chromatography, TLC for short, and these are all the mobile phases. In the first jar, it contained butanol, acetone, water, and ammonium hydroxide in a 50-30-15-3 ratio. Our second mobile phase contained methanol and ethyl acetate in a 1-to-1 ratio. Our third jar contained only ethyl acetate. Our fourth jar contained ethanol and water in a 9-to-1 ratio. And our fifth contained just methanol.
All right, guys, I know it's a little complicated, but bear with us because it's really interesting. So the first result of our diazotization reaction resulted in a negative absorbance spectrum, which indicated that we probably had some sort of problem. Because a negative absorbance spectrum, in this case, indicates that our solution is transmitting more light than it's absorbing, which in this case wasn't possible because our solution was completely clear. So we looked into it a little bit and found out that we had incorrectly set the spectrophotometer in regards to blanking the system and the solvent. Okay. So after fixing the problem, we ran the samples through the spectrophotometer once more. And we found that samples 1 and 2 and sample 3 were very similar in terms of their spectra. There we go. So they both have very similar graphs, for one thing. And they also both have peaks at 230. Around 230. And this indicates that they're likely the same product because they had such similar absorbance spectra. And this is why we only tested one of them for the proton NMR. Samples 4, 5, and 6, on the other hand, was very different. It had 3 peaks. And this indicated that it was a different compound from samples 1, 2, and 3. And that's why we put this one through the proton NMR separately. Our starting material was 2-amino-6-chloropyrene, and it had 5 peaks. This was encouraging because our samples were so different from our starting material that it was likely that we had created some sort of new compound. So we put all of our samples through the proton NMR, and now our literature says that our desired product, which was 2-fluoro-6-chloropurine, would have peaks around 11.97 and 8.65, as you can see here. But our sample had peaks all in between 0 and 4, which indicated that we had not created 2-fluoro-6-chloropurine. The same was true for samples 4, 5, and 6. All of the peaks were in between 0 and 4, so we hadn't created our desired product.
Okay, so there's some errors in our first reaction, and during that we added too much salt, and that was one error. And then,
all right, so we had a couple of errors in our methods. Like, for one thing, we may have accidentally gotten salt into our solution when we were cooling it because we were using an ice-salt mixture to cool down our solution, and probably got salt into there. And during purification, we put in sodium hydroxide to neutralize our solution. But we put in too much, and so we needed to dissolve our solution in water in order to get rid of the excess. For characterization, we incorrectly set the spectrophotometer, which led to a couple of extra steps that may have disrupted the product or any of the necessary reactions.
So the results from our TLC are right here. And so in the first one that we set up, we see that, okay, so in the first, our starting material is to the left, and that has 2 spots. The next that you can see is hypoxanthine, and that has 1 spot. And we wanted our product to have 1 spot too, and look a little different from hypoxanthine, but on our first trial, it has 2 spots, which shows impurities. So after we filtered it out in our methods and results, we created on the right graph, it shows exactly what we wanted. So this is the UV spectrophotometer results, and this is the starting material, 4,5-diamino-6-hydroxypyrimidine, and that's the spectrophotometry for the first starting material. And then here's the hypoxanthine UV spectrophotometer through it, and then there's our product. It's really blurry, but our product has a peak at 274, and that's different from hypoxanthine at our starting, so that means we created something that we wanted.
So in conclusion, for the first method, for the diazotization reaction, we hadn't created our desired material, but that does not necessarily mean that the method doesn't work. It should be retested in order to ensure that our results were not due to error. For the second method, we know that we created a different compound from our starting material, but further characterization is required before we can take any further steps. So there are references. It's only half of our references. We have so many references. All right, so we would really like to thank everyone who has made this experience possible. So first of all, a huge thanks to Xcel Energy for funding both of our research. And we would really like to thank our mentor, Opa Jane, for teaching us with such patience and clarity. I really have no idea how you did it. And I'd like to thank all the FSI staff for guiding us in the learning process and reading over our papers and staying up past, I think, 1, helping us. I don't know how any of us got through that. And I'd like to extend a warm thanks to both of our families who gave us the opportunity to be here. We really appreciate it. And we'd also like to thank everyone here for making this experience fun and learning. So thank you all. We really, really appreciate it.